Let's get straight to the point. Number 1. Garlic. In 1858, Louis Pasteur published an observation that most people have never heard. He documented that garlic juice killed bacteria in laboratory conditions, killed it completely, in concentrations that he described as remarkable. This was before antibiotics existed. Before germ theory was fully established, Pasteur was not studying garlic. He simply noticed what it did and wrote it down. What he was observing was allicin. Allicin is formed the moment a garlic clove is crushed or chopped, a defensive compound the plant produces when its cell walls are breached. It is one of the most potent natural antimicrobial compounds ever characterized, with documented activity against a broad range of bacteria, viruses, and fungi. The problem is that allicin is thermally unstable. Heat destroys it within minutes. The garlic that goes into most modern cooking, diced and dropped into a hot pan, loses its allicin almost immediately. What remains is flavor. What your ancestors consumed when they ate raw crushed garlic was something pharmacologically different. Ancient Egyptian physicians prescribed raw garlic to pyramid workers. The Ebers Papyrus, one of the oldest medical documents in existence, dated to approximately 1550 BCE, contains 22 separate garlic prescriptions. Roman soldiers carried it. When World War I field hospitals ran out of antiseptics, surgeons packed wounds with raw garlic paste because it was what they had. Infection rates in those wounds were documented as significantly lower than expected. The mechanism was not understood then. Pasteur had pointed at it half a century earlier. Aged black garlic adds a second dimension. When raw garlic is fermented at controlled temperature over several weeks, allicin converts into stable organosulfur compounds, S-allocysteine among them, that survive digestion and reach the bloodstream intact. A 2012 meta-analysis in the Journal of Nutrition found aged garlic extract produced significant reductions in systolic blood pressure in hypertensive patients. Different mechanism, different compounds, the same plant, prepared differently, doing something different, which is exactly what your ancestors understood intuitively and what took modern science a century to characterize. Number 2. Ginger. In 2005, researchers at the University of Michigan published a study that received almost no mainstream attention despite its findings. They found that gingerols, the primary active compounds in fresh ginger root, induced cell death in ovarian cancer cells in laboratory conditions, including cells that had developed resistance to standard platinum-based chemotherapy. The researchers were careful about their conclusions. Laboratory findings are not clinical outcomes. But the mechanism they identified, the specific pathway through which gingerols induced apoptosis, was real, documented, and published in a peer-reviewed oncology journal. That a kitchen spice was producing that kind of activity in a laboratory was not entirely surprising to researchers who had been paying attention to ginger's history. Ginger has been traded as a commodity for over 5,000 years. A single pound of ginger in medieval England was worth the equivalent of a sheep, not because it made food taste better, but because people who used it regularly experienced consistent enough benefits that they considered it worth that price. Arab physicians in the 9th century documented it for digestion and joint discomfort. Chinese medicine had been using it for 2,000 years before that. Indian Ayurvedic texts from 3,000 years ago called it a universal medicine. 3 separate ancient medical traditions on 3 continents arrived at the same plant for the same conditions without any contact with each other. The distinction between fresh and dried ginger turned out to matter significantly. Fresh ginger is highest in gingerols. When ginger is dried, those gingerols convert into shougals through a dehydration reaction, compounds with a different molecular structure and a measurably stronger anti-inflammatory profile. A 2015 study in the Journal of Agricultural and Food Chemistry confirmed that shougals demonstrated greater anti-inflammatory potency than gingerols across multiple tested pathways. Your ancestors used both forms because centuries of observation taught them that fresh ginger and dried ginger did not behave identically. The chemistry confirmed what the observation had already established. Number 3. Elderflower. In 1995, a randomized placebo-controlled trial was conducted in Panama during an active influenza B outbreak. Researchers gave one group elderberry extract, the other group received placebo. The elderberry group recovered in an average of 2 to 3 days. The placebo group took 6. The study was published in the Journal of Alternative and Complementary Medicine, and largely ignored by mainstream medicine for the next decade. The mechanism behind those results was characterized later. Elderberry anthocyanins, the compounds responsible for the deep purple pigmentation, were found to bind to influenza virions directly, inhibiting their ability to penetrate cell membranes and replicate. This is a specific, documented antiviral mechanism, not immune stimulation in a general sense. Direct interference with viral replication at the cellular entry point. A 2016 randomized trial confirmed the earlier findings in a larger population. Elderberry supplementation in air travelers reduced cold duration by nearly 2 days and cut symptom severity significantly. The European tradition that surrounded this tree for centuries understood something the 1995 trial confirmed. Rural communities across Britain and Northern Europe maintained elder trees at the edge of every farm and kitchen garden for exactly this reason. The flowers in summer for fever and respiratory illness, the berries in autumn for immune support through winter. Every part of the tree, every season, had a documented application passed from generation to generation without interruption. Then antibiotics arrived. The medicine chest became irrelevant. The elder tree became an ornamental, and the knowledge that a tree growing freely in any garden could reduce a flu's duration by half was quietly reclassified as folklore. The 1995 Panama trial was not discovering something new. It was confirming something very old. Number 4. Rhodiola Rosea. This plant grows above the Arctic Circle, in conditions that should make plant life impossible. Sub-zero temperatures for months, thin rocky soil, intense UV radiation, brutal wind. Most living things cannot survive there. Rhodiola not only survives, it thrives. Viking warriors used it before battle. Siberian hunters chewed it before long expeditions into cold wilderness. Sherpa guides in the Himalayas used it for altitude endurance. These are not connected cultures. They are people in extreme environments who independently discovered that this plant changed what their bodies could endure. In the 1960s, the Soviet government became very interested in rhodiola. They wanted to know if it could help soldiers, cosmonauts, and athletes perform better under stress. They ran classified research programs on it for decades. When those files were declassified after 1991, what they contained surprised Western researchers. Rigorous studies showing measurable improvements in physical endurance, mental performance under stress, and recovery from fatigue. The reason rhodiola works is that it developed chemistry to survive conditions that should kill it. Those same compounds, rosavins and salidroside, help human biology manage stress more efficiently, produce energy more effectively under difficult conditions, and recover faster. This is what researchers call adaptogenic action. The plant does not push your body in one direction. It helps your body find its own balance under pressure. A clinical trial published in Phytomedicine found rhodiola significantly improved burnout symptoms, exhaustion, mental fog, stress reactivity within 8 weeks. Number 5. Tulsi, holy basil. In India, the Tulsi plant has grown in the courtyard of almost every traditional Hindu home for thousands of years. Not as decoration, the Charaka Samhita, one of the foundational Ayurvedic texts, written over 2,000 years ago, classified it not as a treatment for any specific disease, but as something consumed daily to maintain the foundation of health itself. Modern pharmacology spent most of the 20th century focused on crisis intervention. The concept of a daily plant input that prevents the drift toward disease did not fit the pharmaceutical model, which requires a disease state to treat. Tulsi did not fit that model, so it was largely ignored in Western research until the early 2000s. What researchers found when they looked was not one mechanism, but several operating simultaneously. Eugenol inhibits COX-2, the same enzyme targeted by anti-inflammatory drugs like ibuprofen, reducing prostaglandin production and the chronic inflammatory signaling that underlies most modern chronic disease. A 2012 randomized placebo-controlled trial found 6 weeks of Tulsi supplementation produced significant simultaneous improvements in cognitive function, stress response markers, and fasting blood glucose. Not one outcome. 3. Measured concurrently, in a controlled trial, in a plant that has been growing in Indian courtyards for 2,000 years. Number 6. Yarrow, Achilles, the Greek hero of the Trojan War. His name is embedded in this plant's Latin name, Achillea millefolium, because legend says he used yarrow to treat the wounds of his soldiers on the battlefield. The legend is probably not literally true. But the fact that the ancient Greeks connected yarrow to wound treatment so strongly that they named it after their most famous warrior tells you something real about how consistently it was used for exactly that purpose. Archaeological evidence from ancient burial sites suggests yarrow was among the plants placed with the dead, implying its significance predates written history by tens of thousands of years. Roman soldiers carried it. Medieval battlefield surgeons used it. Native American tribes across North America, who had no contact with European traditions, used yarrow for wounds and fever independently, arriving at the same plant through the same observation. Yarrow contains compounds that support the body's natural clotting process, reduce inflammation, and demonstrate mild antimicrobial properties, exactly the combination you would want from a wound plant. It also contains compounds that promote perspiration, making it useful for supporting the body during fever. Today, yarrow grows wild across every American state. Most people mow it down without recognition. They are mowing down a plant that human beings have been using since before recorded history. Number 7. Maca Root. The Inca Empire was built at altitude. The Andes Mountains, where Inca civilization flourished, sit between 11,000 and 17,000 feet above sea level. Conditions that push the human body hard. Low oxygen, extreme cold, intense physical demands. Maca root was central to Inca life at that altitude. Warriors ate it before battle. Farmers ate it through the growing season. Inca physicians prescribed it for energy and stamina. Spanish conquistadors, after arriving in Peru, noted that the indigenous populations used maca extensively and appeared to maintain exceptional physical capacity under conditions the Spanish found punishing. The Spanish eventually began feeding maca to their horses to maintain the animal's health and vitality at altitude. They observed it worked. They kept doing it. The horse documentation is, in some ways, more useful than the human accounts, because horses cannot report a placebo effect. Maca contains glucosinolates along with unique alkaloids called macamides, found nowhere else in nature. Research has shown maca supports energy, mood, and hormonal balance, not by directly introducing hormones into the body, but by supporting the systems that regulate hormone production naturally. Clinical trials have found consistent improvements from sustained daily use, exactly how the Inca used it. Number 8. Hibiscus. 3 civilizations, 3 continents, 1 plant. In West Africa, hibiscus tea, called bissap, has been consumed daily for centuries, served at every gathering from casual meals to weddings. In Egypt, carcadé is so embedded in the culture that it appears on tables the way water appears in American restaurants. In Mexico and Central America, agua de Jamaica is a staple that most families make weekly without thinking of it as anything other than a normal drink. These cultures had no historical contact with each other when they developed these traditions. They arrived at the same plant, the same preparation, flowers steeped in water, and the same daily habit through entirely independent observation. Researchers at Tufts University ran a clinical trial on hibiscus and published the results in the Journal of Nutrition. 3 cups of hibiscus tea daily for 6 weeks produced meaningful reductions in blood pressure readings in adults with mildly elevated levels. The mechanism is the deep red color itself, anthocyanins, that support healthy vascular function while also acting as gentle diuretics and reducing arterial inflammation. 3 separate ancient cultures were drinking cardiovascular support at every meal. They just called it tea. Number 9. Bitter melon. Stay with this one, because what the history of this plant reveals about human observational capacity is genuinely remarkable. Bitter melon has been consumed daily across India, Southeast Asia, China, and East Africa for over 3,000 years of documented history. Traditional medicine across all 4 of these cultures, civilizations with no contact with each other, speaking different languages, operating within entirely different belief systems, used it for the same primary condition, what Ayurvedic medicine called sweet urine disease, what Chinese medicine called Shaoke, what we now call type 2 diabetes. 4 independent civilizations, the same intensely bitter vegetable, the same metabolic condition. That convergence is not folklore. It is one of the most consistent examples of independent empirical observation in the history of medicine. A 2011 meta-analysis in the Journal of Ethnopharmacology found bitter melon extract produced significant reductions in fasting blood glucose across multiple randomized controlled trials. The effect sizes were modest and variable, but the direction of the evidence was consistent and the mechanism now characterized. The deeper story is not about managing an existing condition, it is about what happens to a species that removes bitter compounds from its diet entirely. Bitterness was systematically bred out of domesticated vegetables across the 20th century because sweetness sold better. The metabolic signaling that bitter compounds had been providing to human digestive and endocrine systems across millennia was quietly removed from the modern plate. 4 civilizations noticed what it did when it was present. 1 century of agricultural selection removed it without noticing what it was doing. Number 10. Vitex, or chaste tree. The chaste tree has one of the strangest names in the herbal tradition. It was given by medieval monks who believed that consuming the berries reduced sexual desire, and who therefore valued it greatly for life in a monastery. What they were actually observing, across generations of use, was a plant that influenced the female reproductive cycle in a consistent and meaningful way. Women in ancient Greece used it for menstrual irregularity. Hippocrates prescribed it. Dioscorides documented it. The tradition of use for female hormonal support stretches back over 2,500 years without interruption, which is an unusually long continuous record for a specific application. Vitex works by influencing the pituitary gland, the master hormone gland, in ways that support progesterone balance in the second half of the menstrual cycle. Many of the symptoms commonly associated with PMS are linked to hormonal imbalance in that phase. Vitex gently supports that balance. Number 11. Horsetail. Horsetail is one of the oldest plants on Earth. Not old in the way that oak trees are old. Old in the way that means it was here before dinosaurs. Horsetail has existed, essentially unchanged, for over 350 million years. It survived every mass extinction in the geological record. The plant growing in damp ditches along American roadsides today is structurally identical to fossils from the Carboniferous period. Roman soldiers drank horsetail tea for wounds and bone injuries. Medieval herbalists used it for the same applications. Native American tribes used it independently. The consistent application across cultures that had no contact with each other was connective tissue: bones, tendons, ligaments, skin. For a long time, this seemed like an odd convergence without a clear explanation. Then researchers looked at what horsetail actually contains. Horsetail accumulates more bioavailable silica than any other plant on Earth, and silica, specifically orthosilicic acid, is required for collagen production. Not just helpful, required. Your body cannot complete the structural process that gives collagen its strength without adequate silicon. Collagen is the scaffolding of your skin, your tendons, your bone matrix, your arterial walls. When silicon is insufficient, and it is widely insufficient in modern diets because food processing strips it, your collagen is structurally compromised. A 2016 study found supplemental orthosilicic acid from horsetail significantly improved skin elasticity, reduced nail brittleness, and improved hair strength. The Roman soldiers drinking it for bone injuries were accessing the mineral their connective tissue required. The outcome was predictable once you understand the mechanism. Number 12. Oregano. The oregano in your spice cabinet is not what your ancestors used. The dried, mild, Italian seasoning version that most Americans know is a culinary shadow of wild oregano, Oreganum vulgare, grown in the rocky, mineral-rich soils of the Mediterranean. Greek and Roman physicians used wild oregano for over 2,000 years. Hippocrates prescribed it for respiratory support, and as a natural antiseptic. The ancient Greeks used it for infected wounds. The difference between culinary oregano and medicinal oregano is not variety, it is growing conditions. Oregano grown in rocky, dry, mineral-poor Mediterranean soil produces dramatically higher concentrations of its active compounds because the plant is under stress. Stressed plants produce more defensive chemistry. That defensive chemistry is the medicine. The active compound is carvacrol. It disrupts the cell membranes of harmful microorganisms. Laboratory research has shown carvacrol active against a wide range of pathogens. As a daily antimicrobial support, as a topical application for fungal conditions, and as a natural food preservative, the biology is real and documented. The Mediterranean kitchen that used it freely in every meal was performing low-level antimicrobial maintenance without knowing why it was extending the shelf life of their food. Number 13. Sea buckthorn. The Greeks named it hippophae. It translates roughly as shining horse. The name came from observation. Horses that grazed on sea buckthorn returned with coats that were visibly glossier, thicker, and healthier than horses that had not. The Greeks considered the observation significant enough to make it the plant's permanent name. They then began using it themselves for skin and wound care, following the logic that what visibly improved connective tissue health in animals might do the same in humans. Tibetan medicine texts from the 8th century CE documented sea buckthorn for respiratory health, blood support, and skin. Russian cosmonauts received sea buckthorn oil as part of their standard medical protocol during the Soviet space program, specifically for protection against radiation-induced tissue damage. Between ancient Greece and Soviet cosmonauts, the threads of observation stretch across 2,500 years and 3 civilizations with no shared medical tradition. Sea buckthorn contains something almost no other plant provides: omega-7 fatty acid, specifically palmitoleic acid. Omega-7 is a structural component of the mucous membranes that line your digestive tract, your respiratory system, and your skin. These membranes are your body's first line of defense. When omega-7 is insufficient, and most modern diets provide almost none, those membranes lose their optimal protective function. Sea buckthorn restores the raw material they are built from. The shining horses the Greeks observed were experiencing restored membrane health expressed through their coat. The cosmonauts were getting support from membranes that environmental stress affects first. Number 14. He Shou Wu. This plant requires 2 stories: the one that made it famous across 1,000 years of Chinese medicine, and the one that modern pharmacovigilance added to the record much more recently. Both are true. Both matter. You cannot responsibly discuss He Shou Wu without telling both. The first story begins with the name itself. He Shou Wu translates as Mr. He's black hair. The legend says that an elderly man named He Tian-er, frail, gray-haired, childless, discovered this root growing wild, consumed it regularly, and over time regained his dark hair, his vitality, and his fertility. He lived, the legend claims, to 160 years. The story is almost certainly not literally true, but it has been attached to this plant for over 1,000 years of Chinese medical practice because the observations it encodes, that consistent use of this root, produced visible changes in the signs associated with biological aging, were accurate enough to survive in the record. Chinese physicians classified it as a primary tonic for what they called kidney essence, their framework for what we now recognize as hormonal vitality, reproductive function, and the physiological foundations of longevity. It became one of the most widely used plants in the entire Chinese herbal tradition, consumed by hundreds of millions of people over a millennium. Modern research confirmed real biological activity. Estilben glycosides in He Shou Wu demonstrate antioxidant activity and have been studied for effects on cellular aging pathways. Emodin demonstrates anti-inflammatory properties through NF-κB inhibition. The activity is real. And then the second story. Beginning in the 2000s, case reports began appearing in medical literature documenting acute liver injury associated with He Shou Wu supplementation. Over time, researchers began documenting cases of liver injury associated with certain forms of He Shou Wu. Most cases involved raw preparations, very high doses, or prolonged unsupervised use. Traditional Chinese medicine always used processed He Shou Wu, steamed repeatedly with black bean liquid in a preparation called He Shou Wu, specifically because classical practitioners observed that unprocessed root caused gastrointestinal distress and other adverse effects. The processing was not ceremonial. It was a safety protocol developed through observation of adverse outcomes across centuries of use, refined long before anyone had the vocabulary to explain why it was necessary. Modern supplementation frequently uses raw, unprocessed root because it is cheaper to produce. The 450 documented cases are almost entirely associated with raw preparations. The 1,000-year Chinese tradition used processed root under practitioner guidance. The distinction is not minor. It is the entire difference between a plant with a legitimate clinical history and a supplement that causes liver failure. He Shou Wu belongs in this video because its history is real, its activity is documented, and the warning attached to it is a lesson in what happens when traditional preparation knowledge is discarded in favor of manufacturing convenience.